Hello and welcome everyone. This video is going to cover a primer for the Winogradsky columns. In this lab, we are going to explore the role that soil plays in the Earth's processes of biogeochemical cycling. Now that's a large and complex term. So what is biogeochemical cycling? Let's break it down. Bio means that these processes pertain to living organisms, while geo means earth, ground, or land. And chemical refers to a specific atomic or molecular substance. So, taken together, biogeochemical cycling refers to the complex flow of matter through both living organisms and the Earth's non living systems, like the atmosphere and water. There is a specific chemical reaction that we need to cover that is closely related to biogeochemical reactions, and that is oxidation reduction reactions. Oxidation reduction reactions are a specific type of chemical reaction where one or more electron is transferred between atoms or molecules. As you see here with sodium fluoride, the sodium has an electron seen in blue that is transferred to fluorine and becomes a part of fluorine's electron um, collection. Consequently, sodium becomes positively charged because it's lost a negatively charged particle, the electron, and fluorine gained a negatively charged electron, and so its charge becomes negative. Oxidation reduction reactions is a kind of mouthful to say, so they are usually abbreviated as redox reactions. The most common, simplest redox reaction is the formation of an ionic bond between two atoms, which we see here. Let's take a moment and discuss why they are called oxidation reduction reactions, because understanding what this name actually means can make it easier to remember what these reactions are. First of all, what is the meaning of oxidation? Well, this part of the name derives from the element oxygen. Oxygen is one of the most electron greedy elements, and so when it engages in chemical reactions, it often takes electrons from other atoms or molecules. As a result, the atom or molecule that has lost an electron has been oxidized or acted upon by hydrogen. However, it's important to note that the element oxygen is by no means always the thing taking electrons. It's just so notorious for its electron greediness that it's the namesake that has been used in this case. So, on the prior slide, we saw the formation of sodium fluoride, and fluorine was the oxidizing agent. It was taking an electron from sodium. So that's one half of the equation. What about reduction? Reduction gets its name from the fact that electrons have negative electric charges. So when an atom or molecule gains an electron, it actually experiences a reduction in its electric charge. And because the charge becomes more negative. So that's oxidation and that's reduction. And the two are always coupled with each other because a molecule can't lose an electron without another molecule taking it. If you have an oxidation taking place, you inevitably have a reduction taking place too. In this course, we are strictly dealing with organic molecules when we're talking about redox reactions. So it's going to make it a lot easier to identify redox reactions. And I have two rules of thumb for you for redox reactions. If you have a molecule and before it's reacted in a chemical reaction, um, if the ratio of oxygen in a molecule increases as the reaction takes place, the molecule is being oxidized, which makes sense intuitively. You add more oxygen to a molecule. Oxygen is an oxidizing agent, so we have oxidation occurring on that molecule. Additionally, if the ratio of hydrogen is increasing, the molecule is being reduced. And so we're loading electrons onto that molecule. And that is reduction because electrons are negatively charged. We can flip these two rules on their head here. And with rule number one, if the ratio of oxygen molecules declines, then we would not say it's being oxidized. In fact, it's being reduced. And if the ratio of hydrogen is declining, then the molecule is not being reduced, it's being oxidized. Let me show you an example. Here we have the chemical formula for photosynthesis. 6 CO2 molecules, carbon dioxide, reacts with 6 water molecules, H2O. And in the presence of solar energy, they react to form 1 glucose molecule, C6H12O6, and 6 oxygen molecules, 6O2s. So if we look at the before, the precursor molecule to glucose, that would be carbon dioxide. And you see there's 1 carbon for every 2 oxygens. If we look at the final product, glucose, there's 1 carbon to 1 oxygen. So if we examine our rule number 1, we can see that the number of oxygens has declined, or the ratio has declined. Since it's declined, we know that it's not being oxidized, it's being reduced. Additionally, we can confirm this by looking at the hydrogens. Remember, rule number 2 says if the ratio of hydrogens increases, the molecule is being reduced. CO2 has no hydrogens, and glucose has 12. So we've double confirmed that this is being reduced. So CO2 is reduced. And if we look at water, H2O, we have 2 hydrogens to 1 oxygen. And the final product, oxygen, has no hydrogens. And so again, our hydrogen is being decreased in its ratio. And so we would say it's being oxidized into oxygen. And I want you to notice here that this is a coupled reaction. Carbon dioxide is reduced, so it's gaining electrons to become organic matter. And it has to get those electrons from somewhere. So it gets those electrons from H2O. And if H2O is giving its electrons away, it's being oxidized. These redox reactions are critical to understand when we're looking at biogeochemical cycling. When we look at the reverse process of photosynthesis, cellular respiration, where a glucose molecule reacts with 6 oxygen molecules to become 6 carbon dioxides and 6 waters, we can determine that this is also a redox reaction. Glucose turns into carbon dioxide. It has a decrease in hydrogens, which means it's being oxidized, and it also has an increase in the ratio of oxygens. And an increase in oxygens means it's being oxidized. So we've confirmed glucose is oxidized. Conversely, the oxygen is turned into water, and that increases the number of hydrogens. And so we know that it is being reduced. And again, we have a coupled reaction, a redox reaction. So why are redox reactions so important? Well, we showed how it's related to photosynthesis and cellular respiration, which is part of the biogeochemical cycling of water, oxygen, hydrogen, and carbon. But these redox reactions move stored electrons around, and those stored electrons are our potential energy that gets stored. And we're familiar with this when it comes to electrons moving through a circuit in our electrical systems, but this is how biological organisms do it as well. So redox reactions move energy around, and that energy can be captured to do work for the cell. You can think of those chemical bonds, those electrons being stored in chemical bonds, as the potential energy in a water hydroelectric dam. And when we move those electrons, we are converting that potential energy into kinetic energy. And when you move electrons, that's a redox reaction. The molecule that loses those electrons is oxidized, and the molecule that gains those electrons is reduced. There are two main reasons that organisms do redox reactions. One is to assimilate matter. So if you want to combine different atoms to each other, you're going to be doing a redox reaction. And the other is, again, to store and harvest energy, like we see with photosynthesis or cellular respiration. And so we can always ask ourselves when looking at a metabolic activity, which of these two purposes are being met? Is it done to store or assimilate matter, or is it done to harvest or store energy? So we probably already have a pretty good handle on one biogeochemical cycle at this point, and that's carbon. Carbon is an extremely important element for living organisms. In fact, it's the basis for all of the essential organisms, molecules like DNA, proteins, and carbohydrates, shown here on the periodic table of elements. And we also have specific terms that we use to describe different types of organisms based on how near or removed their energy gathering processes are from that ultimate source being the sun. And that is called an autotroph or a heterotroph. Autotroph literally means self-feeding. The prefix auto means self, for example, an automatic transition, also known as a stick shift, um, as a manual transmission, as a car where you have to change the gear yourself. Troph is a root word that means food or nutrition. So, members of this photoautotroph group, including all plants and algae and certain types of bacteria called cyanobacteria, are able to synthesize their own foods, autotrophs. On the other hand, we have heterotrophs, and heterotrophs cannot obtain their own carbon by using the sun. They have to consume other organic carbon, and hetero means other, and this allows them to assimilate matter and produce energy. And this would include animals, fungi, and most bacteria and protists. In our discussion of cellular respiration and photosynthesis, we looked at how carbon flows through living organisms. Carbon is found in the in the non-living system of the atmosphere in the form of carbon dioxide gas, and photoautotrophs absorb that carbon dioxide and transform it into biological molecules like carbohydrates. This is referred to as carbon fixation, capturing the gaseous carbon from the atmosphere and fixing it into other biological molecules. Then those biological molecules are broken down through the process of cellular respiration if oxygen is available, or other metabolic processes if oxygen is not available, which releases the carbon dioxide back into the atmosphere as a waste product, and the cycle comes full circle. One important thing I want to take a look at here is this chemical reaction. It's a simplified version of the chemical reaction. All organic molecules ultimately have to come through autotrophy, and we typically just represent that as glucose. Glucose is a carbohydrate, and if you've ever wondered what does it mean to be a carbohydrate, well, the term carbohydrate simply means a carbon that has been hydrated with water. And if you look at C6H12O6, it is essentially the equation carbon plus water, or CH2O. If you take CH2O and you multiply it by 6, so you multiply C by 6, H2 by 6, and O by 6, you actually get C6H12O6, the chemical equation for glucose. And that is why glucose and other sugars are called carbohydrates. And we're going to use this abbreviated form, CH2O, in our metabolic reactions later on. Another thing to keep in mind is that plants do not just do photosynthesis, they do photosynthesis and cellular respiration. Yes, they have chloroplasts and they also have mitochondria as well. And so they make glucose when they are photosynthesizing. And when they want to release the energy built up in that glucose, they can break it down through aerobic respiration to release the kinetic energy stored in those chemical bonds through the process of oxidizing glucose and reducing oxygen. Now, energy production in oxygen rich environments is very different than energy production in oxygen limited environments. We described earlier that oxygen is a very electron greedy element, and therefore we can harvest an enormous amount of energy by oxidizing organic compounds like glucose and loading those electrons onto oxygen. We do it in our synthesis of CO2, for example. So there are different zones of oxygen availability in the environment. There are oxic zones, and these are regions where organisms that prefer to oxidize organic matter and reduce oxygen can harness energy, and they can create a lot of energy or harvest a lot of energy this way. It is the most preferred way to do so. Unfortunately, only the surface of Earth is abundant in free oxygen. If you go into the depths of the ocean or into the soils, you're quickly going to decrease the amount of oxygen that is available. And when you have very low oxygen or the absence of oxygen present, we have anoxic environments. And here we can expect organic matter or other elements to be reduced instead of oxygen. So other elements or organic matter are going to have to take those electrons away from in order to produce our energy or release it from the chemical bonds of molecules such as glucose. For example, we can reduce carbon molecules. So that's what fermentation is when we convert glucose into lactic acid in yogurt or ethanol. We're reducing that carbon. Methanogenesis is another example. That's methane is CH4. So there's a lot of hydrogens on one carbon. That's a highly reduced carbon. Um, and that's a type of anaerobic respiration. We can replace oxygen with other compounds as well. So we can take nitrate as well, and we can use that as a oxidizing agent instead of oxygen, NO3. And we can do it with sulfur reduction as well. We can take sulfate or SO4 and we can convert it to hydrogen sulfide or elemental sulfur instead of using oxygen. And so, this typical equation that we see for cellular respiration, when oxygen is not present, we have to find other things to replace it with in the presence of glucose. So, let's take a look at what that looks like. So, here in the Winogradsky column specifically, we're going to be replacing it with sulfur. And so, you can take sulfate and you can do the same thing. You can oxidize glucose with sulfate, or you can do it through fermentation. Um, and doing it through fermentation, we are not going to fully oxidize glucose. So you can see that we get lactic acid and it's not fully CO2. So we don't get as much energy out of it, but it still allows organisms to survive in the absence of oxygen. Here's methanogenesis. So methanogenesis is Genesis means to create. So this is a metabolic process that creates methane by transforming carbon dioxide into methane. And if we look at this chemical reaction, the number of oxygens to carbon have gone down. So this isn't oxidation, this is reduction. And also the number of hydrogen goes up. And so this confirms that we're getting a very strong reduction here. So instead of reducing oxygen in a anoxic environment, we reduce carbon dioxide into methane to make energy. It's a tough living, but it can be done by certain archaea. Methanotrophy is the opposite process here. So, what happens when this methane travels up through the soil or up through the water column and it reaches an area that is oxic or rich in oxygen? Well, there are other organisms called methanotrophs. And again, troph means to eat or food, and they can eat methane essentially. So what they do is they take methane and they oxidize it into carbon dioxide, CH4 into CO2. Sounds kind of familiar to photosynthesis and cellular respiration, doesn't it? Now we're reversing this process of um, of oxidation and reduction. We're going to look at the sulfur cycle. So those other parts of redox reactions we can do in place of cellular respiration. They're fun to look at, but really we're going to be focused on the sulfur cycle here for this lab, and this is what I want you to focus on when preparing um, for any exams or questions that we have on the Winogradsky columns. Sulfur is found naturally in rocks and minerals throughout the world, and it's released into soil and water through weathering. Once in the soil and water, the sulfur gets converted into the form of sulfate ions, or SO4, with a negative electric charge through a variety of biochemical and geochemical processes. Another form of sulfur is gaseous sulfur dioxide, or SO2, and it's also emitted into the environment, but not by erosion of rocks, but by volcanic eruptions, as well as the combustion of fossil fuels. And so we can see how sulfur gets released into the environment here. Here we have SO2, H2S. Um, but once it gets into a biological system here, how do we cycle this nutrient around through the environment? We see as sulfate, it's very useful here. Sulfate can be taken up by plants, then can be eaten by animals, and it can be released as waste, as organic waste. It can decompose and go back in and being stored in the ground. But what we can also do with this sulfate here is we can convert it into reduced sulfur. So deep in the soil where oxygen isn't fully present, bacteria can use sulfate instead of oxygen, and they reduce it. They've released the oxygen and they've added hydrogen. So we know this is reduction of sulfur. And in doing so, they can harvest energy. And sometimes this sulfur moves into a more oxic environment, and bacteria will do the opposite. They're going to take oxygen and oxidize it, and they can release the energy through that method as well. And we're really going to focus in here. So up until now, we haven't had to uh, be too concerned with the subject matter here, but um, uh, you know, there's no need to memorize the nitrate cycle or anything like that. But I want you to be familiar with this core part: bacterial reduction and oxidation of sulfur. Okay, this is really important to us because we also need to be able to generate sulfate as fertilizers. And we release sulfur as well into the air, and we have to have a way that those pollutants can cycle through our systems. Now, as most microbiologists are most familiar with, our culturing techniques are typically limited to the petri dish, seen on the left, and the liquid broth culture, shown on the right. Unfortunately, here, these create a very homogeneous environment. If you look at the petri dish, everything growing on the surface is going to be exposed equally to the surface air. And if you look at the shaker is going to spin those cultures around and everything's going to be mixed very well together. And we will not be able to fully understand how nutrients are cycled throughout an ecosystem. This is where the Winogradsky column comes in. And this particular column here, it's on display at the Harvard Museum of Natural History. But it allows scientists to do very important things. It allows scientists to observe microbial interactions. And the production of Winogradsky labs led to a very important discovery. Up until then, they thought that all autotrophy, all organisms that create their own organic matter, um, they thought that they were all photoautotrophs. And what they discovered by looking at Winogradsky columns is no, that's not true. Some organisms can obtain energy to make their own organic matter through other chemicals. And this is the use of chemical energy instead of sunlight energy to fix carbon into organic compounds called chemoautotrophy. And it's all over the globe. Here we can see these microbial mats. You can find these in a variety of wet, aqueous, terrestrial environments. Here are some more at a geothermal mat in Yellowstone. So this is looking at a hydrothermal vent, and you can see a variety of different redox reactions happening here. We have the brown comes from organisms that oxidize iron for energy. The green comes from green sulfur bacteria as well that can oxidize hydrogen sulfide to fix carbon. And so let's look at this column and we can overlay it with a figure to the right here, which shows three different oxygen zones of availability. We have our oxic zone and our anoxic zone. And in the middle is what we would call suboxic zone or a zone that has suboptimal amounts of oxygen present or a low amount of oxygen present. And this Winogradsky column allows us to make observations of chemical cycling to mimic nature. And our columns are going to be supplemented with sulfur. And these columns can help further our understanding of how elements are moved through Earth's biological systems. To recap, chemoautotrophs are capable of harnessing energy from certain chemicals. An example of this would be bacteria that can oxidize sulfur, reduce sulfur, right? Such as hydrogen sulfide. And by oxidizing it, they can harness energy. This is similar to how some organisms will oxidize organic matter to harness energy, like cellular respiration. And then this energy can be used by the organisms to synthesize organic compounds or autotrophy. We'll see that they can have various features. Colorless sulfur bacteria, they are called colorless because they lack pigmentation. They help to recycle reduced sulfur in this manner in the presence of oxygen. And so they use oxygen to oxidize the reduced sulfur. And like cellular respiration does with organic carbon, this is going to reduce oxygen. But instead of CO2 as a waste product, SO4 is produced, or sulfate. Let's take a look at that Winogradsky column again. Here we have overlaid a variety of different zones, as well as chemical reactions in organisms that exist in the column in those different zones or layers. At the very top is where our cyanobacteria and algae would be found. So these are photoautotrophs. They reduce carbon and oxidize water, synthesizing their own organic carbon. If we scroll to the bottom here, in this anoxic zone, oxygen isn't available and sunlight is not as prevalent down here at the bottom. And so what do we do for energy? Well, we might have an abundance of sulfate present in our ecosystem. And so if oxygen isn't present, we can use that sulfate and we can take our carbohydrate here and we can turn it into CO2. So we can oxidize it. And through oxidation, that means that it's handing its electrons over to the oxidizer, sulfate, instead of oxygen, and that creates hydrogen sulfide. Those are called sulfate reducing bacteria. And a layer above this that is maybe more in the suboxic zone, a little bit less deficient in oxygen. We can have chemoautotrophs, such as green sulfur bacteria, and they can reduce carbon and oxidize hydrogen sulfide. So they take the product of the sulfate reducing bacteria and they can oxidize that into sulfur. And they do this not to gain energy, but to create organic matter. Here we have carbohydrate. Remember, I said that organisms may do these redox reactions for two main reasons, either to harvest energy, such as sulfate reducing bacteria, or in order to create and assimilate their own organic matter, such as green sulfur bacteria. Now we can take that sulfur product and continue to further oxidize it in the presence of oxygen. So we can take hydrogen sulfide and fully convert it into sulfur, and we can use those electrons in order to reduce or fix carbon dioxide. And again, we can do that here. We can take that elemental sulfur or hydrogen sulfide and fully oxidize it, or fully take electrons from CO2, or sorry, donate those electrons here in order to reduce CO2 into carbohydrates here. And this would be done by colorless sulfur bacteria. And so these different processes allow us to cycle nutrients. And we can see the accumulative metabolic activities will create layers where certain nutrients are found. So if we look at the Winogradsky column, there's going to be a high layer of hydrogen sulfide at the bottom. And as we convert that here towards the top, we're going to be converting it into sulfate, not sulfide. So very low concentration found at the top. So we create a layering of different nutrients and a layering of different metabolic activities. We have sulfate at the bottom that gets converted into hydrogen sulfide. So lots of hydrogen sulfide being created because that's how sulfate reducing bacteria are going to make a living, by reducing sulfate. And as we traverse up through the top, we're going to see different strategies where we go from reducing sulfur, and then we're going to start oxidizing it. See, we lessen the number of hydrogens moving up, increasing the number of oxygen. So it becomes oxidized as we move up the column here into more oxic zones. And so in turn, this influences the microbes that are found in these different layers. Different microbes have different metabolic strategies, and we're going to find them stratified throughout our Winogradsky column. For example, here with the purple sulfur bacteria, we can oxidize hydrogen sulfide into elemental sulfur, and that allows us to reduce carbon or turn it into organic matter. And that is called sulfate oxidation. It seems like a lot, but please use this as a guide to answer your pre-lab questions. Now let's go over how we're going to set up our Winogradsky column. First, there's a variety of required materials. Most of these you should have um, in a home or kitchen. So we're going to need, importantly here, we're going to need 4 transparent 1-liter plastic bottles, essentially water bottles. It's important in bold here that they are transparent because we need light to penetrate to them. So you're going to also need to remove any labels that you have on it. And they need to be 1 liter in order to hold enough material. So look at the label before you purchase any water bottles. If it says 500 milliliters or 16 ounces or a half a quart, that's not enough. Those are tiny. We need 1 liter water bottles, or 1,000 milliliters, or maybe you can say 1 quart or 32 ounces. Any of those numbers, and you know you found the right size. If you don't have a funnel that will fit into the top opening there, I recommend getting 5 of these plastic bottles because you can convert one to a funnel. Even if you do have a funnel, the funnel's opening at the bottom might be so small that it's too much work to get it in. But if you use another water bottle and make a funnel out of it, which I'm going to show you in a moment here, it'll be a lot easier, this step of adding soil to each column. You're going to need something to go out and dig your sample, your sediment sample with, like a shovel or trowel. You're going to need a marker to label each column and some tape that you'll put on each column and then write on to create labels with. You'll need water. Now, preferably, I'd say take it from the source if you can, fill up a water jug or something. If not, tap water is going to work just fine for this experiment. And you're going to need something to store your soil in from the location you acquired it from and carry it. So a bucket, some large container, something that's going to be able to hold a whole gallon of soil. And you want to make sure you have at least a gallon of soil or 16 cups. Next, you're going to need a location to keep your Winogradsky columns. And we need a bright windowsill in order to allow photosynthesis to occur. The brighter, the better. And it's going to need to be in an area where it can remain undisturbed. So you don't want it somewhere that is going to get knocked over. If it gets knocked over, you're going to disturb those layers and it's going to ruin the experiment. You're going to need a measuring cup that measures 1 cup in order to measure out the sediment or other additives. You'll need a carbon source to add to 2 of the water bottles. That carbon source that we use is shredded newspaper, could be shredded notebook paper, and you want it to hold approximately 2 loose cups prior to shredding. So if you crinkle up the notebook paper or newspaper into a cup and it's kind of loosely packed in there, that's about how much carbon you need. So make sure you measure before you cut your newspaper. The sulfur source is going to be eggs. You'll need 4 eggs. Specifically, you'll be using the egg yolks. And again, about 16 to 18 cups of sediment, or about 1 gallon. You'll need a camera to take pictures of your experiment. And if you have a funnel, you may not need to get that 5th plastic bottle, although I recommend using that instead of uh, a funnel you just buy from the store. And this is optional, but a spray bottle is going to help you for cleanup a lot more. I do recommend a spray bottle if you have one. So what you're going to do is you're going to locate a sediment source, preferably a wet one. So from a stream, a creek, a marsh, a pond, bay, ocean, beach, or even just a backyard puddle, maybe under where your hose spigot is or something like that, that tends to get a lot of watering, is highly recommended. If you have no options and there's no possible way you can travel to locate any other soil but dry soil, best to use something than nothing. But the wetter that soil is kept, the better the experiment will go and the more success you'll have. You're going to get that sediment collection. You're going to put it in your container. I recommend having a container with a lid. Maybe it's a bucket, and bring a shovel or a trowel and a camera with you. Collect that sample in the container. You're going to want to take a picture of the site. So make sure you get a picture of your sample site there. While you're gathering the soil, save yourself some effort and just gently sift through and pick out any obvious rocks or debris. So anything that's larger than, I would say, maybe even a nickel in diameter, don't put it in your bucket. Don't take it home to set up. And do not use soil from a garden store. Soil from a garden store doesn't actually have, um, to any appreciable amount, sand, silt, or clay. It's mostly just compost material like bark that's been composted, and we don't want to use that for the experiment. It's going to ruin it. All right, so you're going to return back to your home or your lab to assemble the Winogradsky columns. And this is what I meant by making your own funnel. So you're going to add your soil to each one of your columns. And if you didn't have a funnel, or if you prefer to do it this way, which I would, you're going to take that 5th bottle, you're going to cut it in half, You're going to invert it and you're going to tape it to the top opening there. This is wonderful because the hole is exactly as big as possible. And so you're going to be able to more easily fit your soil down the hole if you do use this method. I recommend taping it just to make it a little bit cleaner. And then you can kind of wiggle it off and the tape will remain on the funnel. And you just use it as a collar to reattach it to the next bottle as you add your soil. You're going to label your 4 columns or your 4 bottles. So this is the control. I put a piece of tape and wrote control on it. But you're going to label 1 control, 1 carbon, sulfur, and then 1 that has carbon plus sulfur. I recommend a pair of gloves to keep your gloves clean. And you're going to thoroughly mix the sediment sample, break up any large clumps because you're going to have to mix the soil with water thoroughly and your amendments thoroughly. And if you break up those large clumps before you add it in, the easier it will to do the mixing with your hands than by shaking this water bottle a million times. You're going to add approximately 3 cups. Don't add any more. About 3 cups of sediment sample to each of your Winogradsky columns using your funnel. After you've done that for all of them, the control column you're going to set aside until step number 8. For the carbon column, the one labeled carbon, you're going to shred a 1 cup of newspaper into about 1-inch sizes or smaller and add it to the column. And then you're going to put the lid back on the column. You're going to shake it up well. It's going to be hard. You're going to have to turn it upside down and left and right, shake it all up so that the newspaper is spread throughout the entire column. And then you're going to need to also do that for the carbon plus sulfur column. For the sulfur column, you're going to crack raw eggs and you're going to remove the whites. So if you haven't done that before, you basically pass the yolk back and forth between the 2 halves of the egg until all you have left is the raw yolk, and you can wash the egg white down the drain or cook it and eat it. And then you're going to place the cap on and mix those 2 egg yolks thoroughly. For the carbon and sulfur column, you're going to do the same thing. So now this carbon and sulfur column has both the carbon source being the newspaper and the sulfur source being the egg yolks. Okay, so at this point, each sediment mixture should be filled about halfway to 3/4 of the column with just your soil and your amendments. If any are below this, then you probably didn't add the right amount of soil. So you want to add a little bit more until they're all about the same height. Now you've got to add the water. This is the really tricky stage. It's tricky because you don't want to add too much water, and once you add too much water, you can't separate the water from the soil. So, what you want to do, I recommend, is if you have relatively dry soil, start with a half a cup of water, pour it into your column, then put the lid on tightly and mix it thoroughly. Shake it up really well. Now take a look at your column. See how well that soil is mixed thoroughly. When it's mixed thoroughly, then see how runny it is. You want it to be about the consistency of soft serve ice cream, maybe slightly softer or a little bit more melted than soft serve ice cream. You don't want it to look like a puddle of mud. You want it to still be rather chunky because you just want a little bit of water that overlays the top. You don't want, when all the soil settles, you don't want to have a giant water layer at the top and all of this mud just at the very bottom. Okay. So if it's not there yet, that's fine. Then add another 1/4 cup at a time. Put the lid on, pause, shake it up really well, check it again. Eventually, this whole surface is going to be covered in mud, and you can't tell by looking at the side. So you have to look through the hole at the top and see how mixed up it is. Once you feel like you have that consistency of a soft serve ice cream or maybe slightly melted soft serve ice cream, it's time to finish up the process. So you want to put the column on your workbench or your table and kind of tap it so that you're releasing any air bubbles and all the soil falls to the bottom instead of kind of being stuck in big chunks on the sidewall. You're still going to have a thin coating around your sidewall where it's above in the air, and we need to get rid of that. So the way you do that is you allow the column to set up for about 2 to 5 minutes and then take the column into the sink so you don't get water or dirt everywhere. And I recommend using a spray bottle if available. And you turn the spray bottle on the squirt setting and take off the cap and try and spray off the walls. See this column, the walls weren't sprayed quite well enough because they added too much water. But if you can, try and spray off as much of the sidewall so all the soil rinses off into the bottom here. And so you just have all of your soil at the bottom, a nice little water overlay. And the water overlay can be about 1 to 2 inches is about right. So this is about right. This one's a little too much, and then you're good to go. And make sure you complete this step for all of the columns. And so you should have a setup that looks something like this. You're going to screw the lids back on, but you don't want them on airtight because these organisms that grow in here, they're going to release CO2 as they're oxidizing carbon matter in your soil. So you want to crack the lid. So make sure you go ahead and put them on tight and then loosen them so that air can easily move in and out of the bottle. You do not want a pressurized bottle. So I would say the looser, probably the better. Um, just on there enough that you're keeping insects from getting into your columns. Place all 4 of those Winogradsky columns into a well-lit horizontal surface. So here's a horizontal surface. I'm getting lots of sunlight on these columns. The more sunlight, the more likely your organisms are going to grow because that's a source of energy. Having it just sitting in a room, you might feel like it's well lit in a room, but it's not. It's a fraction of what sunlight's going to provide. So make sure you have it in a well exposed window or outside on a patio or something like that where it gets plenty of light. And over time, it's going to look something like this. You're going to have some green sulfur bacteria, some purple sulfur bacteria, some purple and green non-sulfur bacteria in your anoxic and suboxic zones, some cyanobacteria at the top, maybe even some iron oxidizing bacteria towards the top where they oxidize iron. What you want to do is you've already taken your sample, you took a picture of the sample site, you've brought it home, you've set up all of your columns like this, and you're ready to take your week 0 pictures. Go ahead and take a close-up picture of all 4 columns. Do it from the side, not on the top. I don't want to see the top. I need to see what's going on the sides. Make sure it's clearly visible, the label. This picture is not a very good picture. I use this just to show you where to put them, but because the light is so bright in the background, it's very shaded in the foreground here, and I can't see the pigmentation. So it's best when you take your culture, when you go to take your pictures, move them out of the bright sunlight, take a picture in them so I can easily see the object that you're trying to take a picture of. And each week you're going to do this. You're going to take pictures and you're going to follow the lab protocol. It's going to tell you how to take pictures, where to put them in your document, what to name them, and then what to write underneath them. So you're going to document your journey here throughout all 8 weeks of the experiment. And that will be your final report that you submit at the end of the semester. You cannot submit a final report if you haven't shown me in the very beginning that you set it up in week 0. So make sure you submit it this week so that we can get that credit counted towards your final grade. All right. That is all. I hope that was helpful for your setup. Please reach out to me for any questions. You may get a variety of different products as well. Here's iron sulfide. So I'm really excited to see what your cultures are going to look like. So for the tasks for this week, you need to complete the Winogradsky pre-lab worksheet that I have posted. You need to collect your soil sample and you need to set up your Winogradsky columns and take pictures of it and send and submit your initial setup for your Winogradsky columns. That's all. Thank you all so much. Let me know if you have any questions, and I will provide feedback on your initial Winogradsky setup columns if we need to make any additional changes. So be on the lookout for that. Thank you all.